Hello, my name is James Pope. I'm one of the directors and founders of Turner Pope Investments. We'd like to welcome you all today to the first in a series of webinars that we will be putting on in conjunction with our friends at Vox Markets. Now today we have Dr. Alastair Smith, CEO of Avacta PLC, who will be presenting. As an advisor to the company, Turner Pope were able to secure a good allocation of stock for our clients in the recent £48 million fundraise, as well as previous rounds. We'd like to thank everyone who's taken the time to register and attend today's webinar. It's been a tremendous response and turnout. Now you will be able to ask questions during the webinar using the box on your screen provided. Now, without further ado, I would like to hand over to Alastair Smith, who can update you on the company and the progress that Avacta has made. Thank you.
Afternoon, everybody. Thanks very much for joining me for the investor update for Avacta Group PLC. I'm aware that there's quite a large number of new shareholders, so I'm going to go through a couple of introductory slides just to introduce the company and the core businesses that we're progressing. So the company, as I'm sure many of you know, is divided into two divisions: the Therapeutics division, which is based in Cambridge, and the Diagnostics division in Wetherby in Yorkshire. So the Therapeutics division, it's important to say, is based upon two platform technologies. So that this is not a biotech with one or two drug assets going into the clinic. It's based on platform technologies generating pipelines of assets for our in-house programs and the programs for partners that I'll mention in a moment. So the R&D centre based in Cambridge has around 33 scientists. We have an in-house focus on immuno-oncology, and we'll touch on that later on in the presentation. And we have global partnerships with four key players that you see on the slide there: Moderna, ADC Therapeutics, Daewoong Pharmaceutical in South Korea, and LG Chem in South Korea. On the diagnostics side, as I say, based in Wetherby, we also have the AIM PLC headquarters there. So there's about 40 individuals, about 30 of those in the lab. Again, in-house product development in the diagnostic space, and we'll touch on that in a moment, as well as a large number of partnerships, most of which are not disclosed. But of course, the partnership with Cytiva and Adeptrix around the COVID19 antigen testing are, of course, in the public domain. And the Diagnostics division bases its product development and its partnerships around the Affimer platform. So one of the key messages that I want to get across is, that whilst the COVID-19 antigen testing opportunity has it's shone a spotlight on what the Affimer platform can do, and has obviously changed the valuation of the company and allowed us to recently raise funds to really put the company in a super strong position over the next 3 years or so. Whilst that is the case, there is a substantial core business which we've developed over the last 3 years or so that has the potential to deliver real long-term sustainable shareholder value. And I think it's worth just going through that, those core programs in a little bit more detail. So on the therapeutic side, as I mentioned, the Affimer platform and the Precision platform producing pipelines of drug assets for both ourselves and our partners. So if I start on the Precision platform and just talk through that very briefly. So there we have a number of opportunities to develop safer forms of chemotherapy. I'll expand on that in a moment. But this is a technology that allows us to take a standard form of chemotherapy, and if I can put it crudely, remove the side effects. So the first of those, AVA6000 or Prodoxorubicin, that will be developed into the clinic later this year or early next year. there is, as we'll see on the subsequent slide, a pipeline of other chemotherapies to which we can apply the precision technology, and I'll talk through that pipeline on a subsequent slide. The precision technology and the Affimer technology together allow us to develop a completely novel form of drug conjugate. So this is where we combine Affimer immunotherapies with precision chemotherapies to deliver both of those types of therapy to the patient in a single drug molecule. And again, I'll talk about the lead program there and the potential for a pipeline of these drug conjugates based on the PD-L1 immunotherapy that we've developed over the last couple of years. On the Affimer side of the therapeutic development, we have quite a modest, at the moment, although the funds we've just raised, of course, allow us to expand on that, and I will talk about that at the end of the presentation. Quite a modest immunotherapy pipeline, but we've established those 4 collaborations, one with LG Chem, which is a significant partnership around a multiple array of targets. Moderna, again, a multi-target deal. They've just exercised, or last year exercised, an option to take one of those sets of Affimers against a particular target into clinical development. And ADC Therapeutics, which is a new collaboration around conventional drug conjugates. Those 3 partnerships together have the potential to deliver more than $500 million worth of clinical development milestones and obviously royalties on future sales. The partnership with Daewoong is slightly different. It's a joint venture around stem cell therapies, which is Daewoong's expertise. But what we've demonstrated is that we can, with Daewoong, engineer stem cells, which are used for regenerative medicine, for tissue regeneration. We've engineered the stem cells to manufacture and secrete or emit Affimer anti-inflammatories and other Affimer drugs when the stem cells are in situ in the tissue. So this really is a next generation of cell and gene therapy, and it's a joint venture which we own 45%, based in South Korea. So hopefully that gives you a rounded picture of not just the ongoing current programs, but the potential for expanding that pipeline and hitting some of that untapped potential that the company has on the therapeutic side. We'll say more about that on subsequent slides as we talk through the presentation. On the diagnostic side, there's been a focus for the last 18 months to 2 years or so on infectious diseases, therapeutic drug monitoring, point of care rapid testing. So the work we're now doing with Cytiva around COVID19 antigen testing, that this is not a sudden pivot towards infectious disease testing and lateral flow testing. It's something we've been focused on for quite a while now. I mean, it is a perfect example of what the Affimer platform can do: quickly generate reagents against a new diagnostic target and develop those diagnostic tests quickly. So, as I say, it's shone a spotlight on the company and the technology and our capabilities, but it really has allowed us to raise funds in order to access some of the untapped potential that we have in the business. Okay, so I will focus now for a few minutes on the COVID-19 antigen test. Obviously, it's extremely topical. And what I want to do first is just talk through the difference between the 2 types of tests. Have I got coronavirus right now, or have I had coronavirus in the past? So if we just think about have I got coronavirus right now, the conventional way of testing for that at the moment is to use PCR testing, which detects the genome, the DNA, or in this case RNA, inside that virus particle. And that obviously works well. It's not perfect by any means, but it works. And the limitation is the fact that you have to send a sample away to a laboratory. It needs a piece of laboratory equipment. And it takes some time to get that turnaround and get the results back, which could take several days. So if we are to be in a position where we can screen very large numbers of people in the community, then PCR testing is far from ideal. And so one way of addressing that problem, possibly the only way of addressing that problem, is to develop rapid point of care, what are called lateral flow tests. So these are test strips, rather like pregnancy test strips, where you get a result in minutes, one red line or two red lines to indicate a negative or a positive. And that sort of test, using saliva as the sample, because if we're going to roll this out for consumers to use, then nasopharyngeal swabbing, so swabbing your own throat and nose, is going to be very challenging for people. So saliva is probably the ideal sample for getting these rapid results in minutes in a way that a lay user as well as a professional can use. And what we have developed over the last few weeks is Affimer reagents that can detect the spike proteins on the outside of that virus particle. So those are specific to the SARS-CoV-2 virus spike protein, and those Affimer reagents do not cross-react with any of the other SARS, MERS, or other closely related coronaviruses that are out there endemic in society. So we have some highly specific Affimer reagents against the COVID-19 antigen. And we are partnered, as I'm sure many of you know, with Cytiva, or formerly GE Healthcare Life Sciences, to develop those lateral flow test strips. And I'll give you an update on progress in a moment. And then just to complete the picture, so you can put everything in context, the type of test that tells you whether you had coronavirus in the past is being referred to as an antibody test, or more correctly, a serological test. And the reason that doesn't give you an immediate result as to whether you have the infection right now is because it takes a week or two, 10 days at least, to build a substantial number of antibodies that your immune system creates against the infection. So after a week or two, raises a sufficient amount of antibody response to the infection to be able to detect that in the blood. And so that gives you a useful test to show whether you've got the antibodies and had the infection in the past, and hopefully have some degree of immunity. But it doesn't tell you, within that first week of infection, when people are most infectious, whether you have the infection or not. So that's the difference between antibody serological testing or antigen testing, which is what we are developing with Cytiva. Okay, so as I said, it's back in April now, we announced that we had developed a large number of Affimer reagents against the COVID-19 antigen spike protein. And that allows us to develop the type of lateral flow test that we've just been talking about, and also a number of other different types of immunoassay. And we'll mention in a moment the Adeptrix collaboration that we have, and we are actively working with other potential partners to get those Affimer reagents into their product development pipelines as well. So we're working with Cytiva to develop a saliva-based rapid test strip which will detect the virus spike protein. We have, a few weeks ago, we shipped materials to both Cytiva and Adeptrix and made very good progress in both of those developments. And we will, by the end of June, have prototype tests from Cytiva to test both with model samples and with patient samples. And of course, that's an important milestone when we can demonstrate how well the tests work. Just to give you an idea of the timeline following that, which obviously, under normal circumstances, these types of diagnostic developments take a year. And of course, we're compressing that into a much, much shorter timescale, a matter of weeks, which has its own challenges and adds to the risk to some degree, because of course we have to run things in parallel, but clearly that's imperative in the current circumstances. So once we have those prototypes, and I've been asked the question before, and we have a very high level of confidence of having those prototypes by the end of June. The performance of those we can then evaluate and look at transfer into manufacturing. We're putting those manufacturing partners in place in the UK right now. Those manufacturers will then produce what are called validation batches, so batches of the product to allow us to carry out the clinical validation, which is the study where we run patient samples, and that's clearly at the point where we get quantitative data on the sensitivity and specificity of the test. That data allows us to get regulatory approval and then CE mark and launch the product, as I've said many times, as soon as we possibly can during the summer. Okay, so the other collaboration we've put in place with Adeptrix is to really, it's at the other end of the spectrum in terms of the type of test. It's a high-end laboratory test to use the installed base of what are called mass spectrometers in hospitals that are not currently used for COVID-19 testing. Adeptrix can run up to 1,000 samples a day on a single machine. So this would really add significantly to the capacity for COVID-19 testing in hospital labs. The type of assay they have they develop is called a BANS assay. And very recently we announced that we had a working prototype. So we've now evaluated that prototype using model samples, and we'll be moving imminently to testing that with patient samples in both the UK and the US before going through a very similar process of manufacturing, clinical validation and regulatory approval. And I think I've said many times, I think this could be a slightly quicker development. So again, looking to have a product to market during the summer. So in both cases, we're working hard to compress those timescales as much as we possibly can to get product ready for market in a very, very short timescale compared to normal diagnostic development times. And from the point of view of the go-to-market strategy with Adeptrix, we're also in the process of securing commercial partners who are likely to be the mass spectrometry instrument manufacturers to distribute the BANS assay product. Okay, well, let me move on because, as I said right at the start, it's really important that we get across to everyone listening and the new shareholders that we have the real value in this business, in the therapeutic and broader diagnostic pipeline. So I wanted to spend 5 or 10 minutes just talking through the lead therapeutic assets. And the first of those is the lead precision prodrug. So we refer to that as AVA6000 or Prodoxorubicin. And what we have done is with the precision technology, we have taken a generic chemotherapy. So this is doxorubicin, which has been around for very many years, is a standard of care for advanced soft tissue sarcoma, and yet it has really very serious cumulative cardiotoxicity. So the damage to the heart with patients on doxorubicin limits their treatment to maybe only 6 cycles, and many patients don't manage 6 cycles. And nevertheless, this is a $1 billion generic drug. And using the precision technology, what we can do is modify doxorubicin so that when the patient is given the Prodoxorubicin, AVA6000, it's effectively inactive until it gets into the tumor by the normal circulation, and then it gets activated by an enzyme in the tumor. And that enzyme is highly overexpressed. There's a lot of it in the tumor and not in healthy tissue. So this is a way of taking a chemotherapy and making sure it's only activated in the tumor, and therefore sparing healthy tissue from the damage caused by the chemotherapy. So just to illustrate that in the data we see on the right-hand side there in a mouse model. So let me just talk you through that. So on what we see is a graph of the amount of active doxorubicin drug in both the heart and the tumor in a mouse xenograft model of cancer. On the left-hand side is the concentration of normal, when the animals are treated with doxorubicin, at 2 milligrams per kilogram. So that's the dose given to the animals, and it's the maximum dose you can give. And what you see is the same amount of doxorubicin in the heart and in the tumor, which is exactly what you would expect. You give doxorubicin systemically, it goes everywhere and appears in the heart to the same level as the tumor. And that's the issue, that you get damage to the heart in the same way as you get the damage to the tumor cells. On the right-hand side of that graph, what you see is when the animal is dosed with AVA 6000 or pro-doxorubicin, but notice it's 6 times the normal dose, so it's 12 milligrams per kilogram of AVA 6000, which remember is inactive. And only when it goes through the tumor does it get activated to become the active doxorubicin drug. So what you see on the graph there is at 6 times the normal dose, you start to see a little bit appear in the heart. That's because there's a very low level of that enzyme in the body, but you see a little bit beginning to appear. But at that point, you've got 18 times more of the active chemotherapy in the tumor compared with the heart. And that grey arrow shows you what's called the improvement in the therapeutic index that we can achieve by using the precision technology. So we can take a generic drug and we can quite dramatically improve the distribution of that active drug in the tumor compared with healthy tissue. So just thinking about the timeline moving forwards, so there has been some delay just broadly because of the COVID-19 pandemic, and of course hospitals are not running clinical trials, haven't been running many clinical trials for a period of time now. We expect that to come back closer to normal in Q3 and certainly in Q4. We therefore anticipate our regulatory filing in the UK in Q3, and then dosing patients in a very late Q4 or probably more likely very early in Q1 of 21. And some of the proceeds from the placing that we've just completed will allow us to also file an IND, which is the equivalent regulatory submission in the US. And we have lined up already and will line up further clinical trial sites in the US. That's an important commercial and clinical step to have the IND filing as well. So that improvement in tissue distribution and the sparing of the heart from damage by the doxorubicin, you would expect to see that reflected in the effect on the tumor, because it's doxorubicin. We know the drug works. We know it's effective. It's the safety that's the issue with most, if not all, chemotherapies. So here you see on the left-hand side of the slide, here you see the tumor size at 60 days following dosing. And the yellow bar shows you the control group of animals, so the tumor with no treatment has grown very large. With doxorubicin, with the pink bar, you see, again, a large tumor. And on the right-hand side with the 2 blue bars, which are 2 different doses, 9 milligrams per kilogram and 12 milligrams per kilogram of the pro-doxorubicin, you can see that the tumor is very much smaller. And of course, that's reflected in the survival of the animals, which I guess couldn't be more black and white. Treated with doxorubicin at 60 days, all the animals had died, and at 60 days, all the animals were alive when treated with pro-doxorubicin. So that gives us obviously a great deal of confidence going into the phase 1 study in humans. And if we can recapitulate that data in a human trial and show that we can dramatically improve the safety and tolerability so that patients can be treated for much more than 6 cycles, then the outcome will be, is expected to be much better for patients. So that will not only generate a valuable asset, AVA 6000 pro-doxorubicin asset on its own, but the point I made earlier on is that we can apply that precision chemistry, the way in which we can activate the chemotherapy just in the tumor, we can apply that to a range of other chemotherapies which have serious side effects. And I've just given a couple of examples in more detail here. I've chosen Velcade because in the pipeline, that is the next most developed prodrug form of a chemotherapy that we have, and we refer to that as AVA 3996. So Velcade is coming off patent in 22 in a couple of years. It's only approved in multiple myeloma because of some, again, serious dose-limiting toxicities. And as I say, coming off patent in 22. So we have developed that to the point of early stage animal models of cancer. We've looked at that in a pancreatic cancer model, and whilst it's very early stage data, so I'm not going to overplay it at this stage, that looks very promising and performs as well as the standard of care in pancreatic cancer. So that can now, with the placing proceeds, be developed preclinically and towards the clinic. A second example, good example, is paclitaxel, which is one of the taxanes. Now that's an even bigger market and growing very rapidly because of the increased detection rates for prostate and cervical cancer. But again, this has very serious toxicities, particularly peripheral neuropathy. And so again, if we can use the precision chemistry to reduce the side effects and improve the efficacy of paclitaxel, there's a huge opportunity to be exploited there. On the right-hand side of the page, you talk a little bit about the potential economics of that. We are a relatively small biotech. We can't develop a pipeline of 10 of these ourselves. But of course, with the data from AVA 6000, successful phase 1 data, with a pipeline of preclinical assets, there's every possibility of some significant licensing deals, which would involve clinical development milestones as well as royalties on future sales. And that list of chemotherapies that I put there, we have in fact made all of those with our partners at Tufts University. Although Velcade, Pro-Velcade 3996 is the only one that we have any substantial data on, all of those have been made, so we know there's a synthetic route. So again, just trying to build out the picture of Avacta for many of those new shareholders. This is a substantial opportunity on its own, let alone the next step, which is to combine those types of chemotherapies with immunotherapies. And very briefly, without going into a lot of the science, it's very clear now for a number of years, large pharma and biotech have been combining chemotherapy with immunotherapy because that gives a better outcome for patients. So that's delivering the chemotherapy with one IV and then following that with a second IV with the immunotherapy. The ultimate combination of these two types of drugs is to put them into a single molecule, because then you have a single drug that you give with one IV, which from a healthcare economics perspective is very attractive. And we have the opportunity to do that. And the precision chemistry goes in the linker that holds the chemotherapy and the Affimer immunotherapy together, so that when that drug conjugate, as it's called, goes into the tumor, the precision linker is cut and the chemotherapy is released, and you get the active chemotherapy in the tumor. What then happens is the chemotherapy will kill cells in the tumor microenvironment. And if you choose the chemotherapy correctly to have what's called a very pro-inflammatory, a way of killing cells that really attracts the immune system into the tumor, then the immunotherapy can support that immune response in attacking the cancer. So it's the synergy between the chemotherapy and the immunotherapy that is critical here and should give better outcomes for patients. I think it's worth just sharing our excitement about some very recent data. So these are animal models of cancer, using the first two of these drug conjugates that we've developed. So this is a colorectal tumor model called a CT26 model, which is generally well known as a difficult model to get a good response, to get the tumor to respond well to drugs in this model. And what we see here is in the left-hand panel, we see the size of the tumor over a 20-day period or so in the control set of animals, so no treatment, and you can see the tumor grows quite rapidly. In the middle panel, what you see is the animals treated with the drug conjugate that we've just described with a PD-L1, an anti-PD-L1 Affimer, which supports the immune system by blocking a pathway that the tumor uses to hide from the immune system, combined with a chemotherapy called IDASH or Valbopro. And what you can see immediately from that middle panel is there is quite a dramatic effect on the rate of tumor growth. And most interestingly, 30% of those animals, the tumors go into complete regression, so the tumors disappear. So that's because of the synergy between the chemotherapy, which kills cells in the tumor mass, which causes a big inflammatory event to recruit the immune system, that is then supported by the PD-L1 Affimer. So that really reflects the mechanism of action that we've been talking about. On the right-hand side, the right-hand panel, at the moment that's an undisclosed Affimer. We're filing some IP around this particular drug conjugate before we talk about that publicly. But what you can see again is a dramatic reduction in tumor growth rate in most of those animals. And in this case, 60% of the animals, the tumors go into complete regression. So that gives us a great deal of encouragement that this novel drug conjugate, which we've patented jointly, it was a joint invention with Tufts University, the concept of having that synergy in the tumor microenvironment in a drug conjugate, that is a proprietary drug modality to Avacta. So that gives us a great deal of encouragement that could make a significant difference to cancer patients. Okay, so just 2 minutes to talk through the partnerships that we have in place. I'll do this very quickly. So I mentioned LG Chem. That's a deal, a headline value of $310 million so that's a deal we signed about 18 months ago, 15 months ago, with $2.5 million up front, around $5 million that we should see over the next 12 or 15, 18 months or so, preclinical milestones. And then royalties on future sales. All of these partnerships, all of them pay Avacta's full research costs. So all the work that we do for these partners is fully funded by the partner. The next key milestone would be delivering Affimer candidates against the second and the third targets that LG selected. So we're now running three programs in parallel with them. And as I say, there's potential for those preclinical milestones to be paid over the next 15, 18 months or so. And LG have publicly said that they will file the first IND around the Affimer programs in 2021. The deal we did with ADC Therapeutics, which is a Swiss biotech that's very recently, the last couple of weeks, listed on NASDAQ, heavily backed by AstraZeneca and with board representation. So ADC Therapeutics has a license to some chemotherapy warheads for conventional drug conjugates that they licensed from AZ. Again, we're doing fully funded development partnership with them, providing Affimers for them to produce drug conjugates instead of using antibodies to produce those drug conjugates. And we signed that deal back in September, October last year. We're now working on the first target for ADC Therapeutics. And the next milestone will be transferring those later this year or early next year to ADC Therapeutics to build into drug conjugates. I'll deal with Moderna first. So Moderna was the first therapeutic partnership that we put in place in 2015. And that really catalyzed a fundraising and allowed us to begin therapeutic development. So, prior to the Moderna partnership, we had no therapeutic development at all. So in the last 5 years, we've gone from this Moderna agreement to the programs and partnerships that we've been talking about. So Moderna last year exercised one of their options to take some of the Affimer lead molecules against one particular target into clinical development. And when they file an IND, which is obviously entirely up to them, we don't control that program, but if and when they file an IND, then we will see a payment, and obviously that's that's an announceable event as far as we're concerned. Daewoong Pharmaceutical, as I mentioned earlier on, is slightly different. That's a joint venture. So we've established a joint venture in South Korea with Daewoong Pharmaceutical, which is, I believe, the largest South Korean pharma company by revenue. And that is focusing on stem cells, mesenchymal stem cells. So these are tissue regeneration and anti-inflammatory therapies. And what we have done, as I mentioned earlier, is demonstrated that we can cause the stem cells to manufacture and secrete Affimer drugs when the stem cells are in situ in the tissue. So that allows the Affimer drug to be chosen to perhaps have an anti-inflammatory effect, really giving an extra level of therapeutic power to the stem cells. So very much a next generation of stem cell and gene therapy. So we are working very hard with Daewoong. They are very keen to make progress very quickly. We've started on 3 targets for them, and we expect to be transferring the first of those sets of Affimer reagents to AffyXL, the joint venture in South Korea, later this year and into early next year for incorporation into those stem cells and preclinical development. So finally, just to sort of wrap up, obviously everybody will be aware that we have just closed a placing and a very heavily oversubscribed placing. We intended to raise around £25 million. It was multiple times oversubscribed. We took £48 million gross, and the use of proceeds, net proceeds here, is obviously split between the therapeutic and the diagnostic businesses. So just to talk you through that very quickly, with regard to diagnostic business, clearly working capital for the COVID-19 antigen testing opportunity, absolutely essential that we have that working capital to be able to carry out the manufacturing scale up, the regulatory, the validation of regulatory process, and expand the facilities and team within the company. I mean, it's essential now that we add some senior expertise around product management, regulatory affairs, more commercial bandwidth, and so on. Also, a good proportion of that £10 million will be focused on accelerating the rest of the diagnostics pipeline, which we haven't had a chance to talk about today, but that pipeline leading to further commercial partnerships. The bulk of the investment over the next 2.5 to 3 years will be in the therapeutic part of the business. And let me just talk you through very briefly the use of proceeds. So first of all, addressing that latent potential in the precision pipeline. So really unlocking that pipeline and developing preclinically several of those. So the Velcade and paclitaxel we talked about, and oxaliplatin are at the front of the list. Expanding the Affimer immunotherapy pipeline, and the strategy here is to take the PD-L1 inhibitor that we've developed right to the point of having a clinical candidate, and to now add to that to produce bispecific candidates by making fusion proteins. There isn't time to go into that, the science behind that now, but that's a relatively rapid process to generate those clinical candidates. And we're looking in particular at bringing forward a PD-L1 TGF-beta receptor trap or inhibitor and a PD-L1 cytokine bispecific. So to develop all of those preclinically and to file IND for one or more of those Affimer immunotherapies, so the lead drug conjugate that we've spoken about and a bispecific, and one or more of those precision prodrugs. Clearly, obtaining data for the Affimer platform in humans is essential, as it has been for a while. We feel as though developing a PD-L1 monotherapy alone, given the time that's elapsed, we had to pause that program last year because of a lack of resources. we strongly believe that there is much greater value to be delivered to shareholders by now developing that PD-L1 antagonist as a bispecific than taking it through as a monotherapy. But essential in this next period is to deliver that human data ourselves and with partners, of course. You know, Moderna potentially, LG very clearly made a statement about getting into the clinic in the next year or two. And finally, I mentioned earlier that the UK clinical trial for the first of those precision prodrugs is fully covered by our current cash reserves, but we will use some of the proceeds of the fundraise to file an IND in the US and look at clinical development in the US as well. Okay, so in summary then, I think the key points that I've tried to get across are, out with the COVID-19 antigen test, which clearly can be transformational on its own for the company. There is a huge amount of value to be unlocked with the funds that we've just raised, that we've developed over the last couple of years. And from a therapeutic perspective, I personally think important to get across the message that this is based on platform technologies, not individual assets with that sort of binary risk of failure that's associated with them. We've got many value inflection points over the coming months and years, but probably the two most significant are the launch of COVID-19 antigen testing products and that phase 1 trial for AVA 6000 prodoxorubicin, which could add huge value to the whole of that pipeline of chemotherapies. As I've discussed, we've got a number of very good partnerships in place that really validate the technology and the approach that we're taking. And we are now fully funded through to 2023 to scale up the diagnostic business for the COVID-19 opportunity, expand the other diagnostic pipeline, and build the immunotherapy and chemotherapy pipelines alongside that. Thank you.
Alastair, thank you ever so much for that presentation. We've
Pleasure.
been monitoring a lot of questions that have been coming in while you've been doing that, and we've tried to select a few that we thought people would want to hear. To start with, there was a bit of news last night about a US investor which has taken over a 3% stake in the company. Could you maybe update us on exactly who that is and how that came to be?
Yeah, of course. Well, over the last 9 or 12 months, I've been doing quite a bit of investor relations work in the US, primarily New York and Boston. And Ruane is a super shareholder to have on board. I've met with them numerous times over the past 9 months or so, and of course with many others. So we've built the story in the US. And got quite a following now, and very, very pleased that Ruane have come on at this particular placing.
With that in mind, people are asking: is there a possibility that Avacta will look to obtain a US listing near term?
Well, I think I'm sure I've said publicly on several occasions that, particularly for the therapeutic activity, the sort of scale-up funds required to run multiple clinical programs in parallel, that's reasonably challenging to access those sorts of scale-up funds in Europe. So yeah, I think one of the strategies we can certainly explore is a US listing to be able to access those scale-up funds.
Okay. Another question that we have: do you see any regulatory interruption in approval of the lateral flow test diagnostic for coronavirus? Does the management expect there to be that to be hindered at the back of a queue of applicants all knocking on the FDA's door?
Well, I mean, clearly regulators are extremely busy at the moment. I mean, they're busy anyway, but there's absolutely no reason whatsoever why Avacta should be at the back of any queue. So, I'm sure listeners are aware there are now accelerated regulatory processes in Europe and the US for professional use of tests. But we're getting into that process now. So as soon as we have news, we'll update the market as we go through clinical validation and into those regulatory processes. No reason at all why we should be at the back of the queue.
Yeah. I know you've touched on the placing, but obviously the recent £48 million fundraise provides significant cash resource. The question we have here says, given the partnership with Cytiva, obviously partnered out, does Avacta appear to have sufficient funds already to take AVA 6000 through to phase 1 clinical trials? Can the management detail exactly how they see that the money raised will be spent over the next 2 to 3 years? So sorry, I know you've already touched on that to some extent, but how is it
So I think I
expected to be
touched
spent?
on that in some detail on the penultimate slide. So exactly as I said on the penultimate slide, it's about working capital for the COVID-19 test, expanding the diagnostic facilities, growing the diagnostic pipeline of our own products, growing the preclinical and clinical pipelines in the therapeutic business, filing an IND as well as a CTA for AVA 6000 and so on.
We do have one question here that talks about your last presentation that you did, and it says during the last presentation you did touch on the fact that the company was funded through to 2021, 2022. It says, do you regret that statement? And is Avacta likely to be a takeover target in the future?
No, well, I don't quite know what that means by do I regret that statement. So that statement was made at the time. The funds we had with the plan we had took us through 21. So that was a situation where we had focused our resources very tightly on AVA 6000 and our partner programs. And now, fortunately, we have been able to raise substantial capital to be able to unlock the pipeline of things that we've simply not been able to to get moving hitherto. So I mean, the statement was correct and is correct.
Alastair, we understand the potential that you have with what's happening. How tricky are Affimer molecules to mass produce, given the anticipated demand for the antigen test? And does the board expect to retain all such production in-house?
That's a really good question. So, I mean, it's one of the major advantages of the Affimer technology. I mean, it is really easy to manufacture. So, right now, for the foreseeable future, we will be able to keep that manufacturing in-house, and part of the use of the proceeds here is to expand those manufacturing capabilities. But in terms of what's called a tech transfer, to transfer that manufacturing capability out to a contract manufacturer, is really straightforward. It's it's a very, very straightforward bacterial expression system.
Alastair, just one more question. Someone's asked here that they expect news on a number of fronts, as previous RNSs have alluded to. What can shareholders expect in the coming weeks and months from Avacta?
No, absolutely. I mean, lots of anticipated news flow. So, we're obviously all very keen to get those prototype devices from Cytiva, so we can test those with patient samples. We'll certainly speak about that when it happens. We're getting manufacturing partners in place for the lateral flow test. We'll clearly be talking about the clinical validation of both that test and the Adeptrix test products. I've said multiple times again today that we're looking to put other commercial partnerships in place for different types of COVID-19 test development and distribution. There's the whole of the rest of that diagnostic pipeline other than COVID-19, which we've not really had a chance to talk about today. But I think at the prelim roadshow, I said that the most advanced of those, 5 of those are now in licensing discussions, having fully completed successfully the evaluations. Then there's potential for a therapeutic deal around the COVID-19 Affimers to inhibit the spike ACE2 interaction, and that, just as an aside, will be enhanced would be more likely if we can show that those Affimers also actually stop the virus entering human cells. So we're working on that. There's a therapeutic pipeline of licensing opportunities and partnerships, which should also generate news flow over the coming months and 12 months. AVA 6000 is going into the clinic, patient recruitment, dosing first patients, progress with our partner programs, getting candidates ready for our partners to take into the clinic, preclinical milestone payments. I mean, there is endless potential for news flow over the next few months.
Alastair, obviously congratulations on the progress you've made so far, and thank you for taking the time to speak to us today. We'd obviously like to thank everyone for attending the webinar, and if you'd like to learn more about Turner Pope and the services we provide, please do visit our website, turnerpope.com. Thank you, Alastair.
Thank you, my pleasure. Thanks, James. Thank you, everyone.